Thank you very much. Okay. So good morning, everybody. Now we're back to our series of lectures on cell biology. These are our final lectures on cell biology. Then what's going to follow up is majority of biochem, genetics. So today we're going to talk about the cell cycle control, and we're going to discuss some molecular basis for occurrence of cancer. So first, we're going to talk about cell cycle. So some of this stuff about cell cycle you already learned before in your undergrad. So we'll be going rather fast through them. But then we're going to discuss about cell cycle control, which might be new information for you. So you know that organisms are formed by the cells who all have specific functions. And we know that whenever an organism needs to grow new cells or replace old cells, it needs to stimulate its cells to divide. Okay? And this process is known as cell proliferation. We know that most of our cells in our body, somatic cells, are actually made from division of existing cells. And we know that cells, whenever they divide, the mother cell will then divide into two equal daughter cells through this process. Now, majority of cells in our body actually is going through turnover. I don't know if my colleagues mentioned to you, but every 2 or 3 weeks, you're a completely new person, okay? Because old cells will die and they will be replaced with new cells. Okay, so again, some old cells will die. Some will actually be just shed off, like the ones that I'm going to talk to you in next block. In the GI, a lot of cells will be shed off due to the process of movement of content through the GI that will scrape off the cells from the surface of your intestines. So some cells will die, some will shed, and they will be replaced by new tissue-specific cells. Now, please understand that some cells do not have turnover. These are known as permanent cells. And for example, good example are neurons or cardiac myocytes. They are not going to be replaced. They're usually replaced by the tissue of lower embryological origin. So what's going to happen is cardiac myocytes will be replaced by connective tissue. Okay? So that is the characteristic. But there are some cells that have very high turnover rate. You're producing tons and tons of red blood cells every day in your body to replace the old ones and damaged ones that are destroyed by your spleen, by reticuloendothelial system. So, you know, in order for cell to split into daughter cells, it needs to do this. It needs to completely copy and all its content. So all the content has to be doubled. The genetic information has to also be contained in chromosome, has to be copied. And then also organelles and filaments have to be copied and transferred all the way to daughter cells. So everything in the cell has to be doubled before the cell divides. So this is a cell cycle. And you heard these are major phases of cell cycle. So broadly, cell cycle is divided into 3 large phases. Into interphase, which is here, blue area. It actually is a most extended area of the cell cycle, or an extended phase of the cell cycle. So it's characterized by cellular growth and DNA synthesis. And it's further divided into G1 phase, S phase, and G2 phase. And we're going to talk about each of these phases again. The next portion is this green area, that's mitosis. It's actually initiation of cell division. It's divided into 5 subphases. So 5 phases under the mitosis. And these are prophase, prometaphase, metaphase, anaphase, and telophase. We're going to mention all of these. And then the next process is cytokinesis, which is the actual division, not of nucleus, but formation of 2 separate cells. Okay? So that is the characteristic of the cell cycle. So we know that interphase is characterized by the cellular growth and also by DNA synthesis. That's the big process. And it's very essential for the duplication of cellular material from mother into the daughter cells. One interesting fact, most of the cells in your body are actually in interphase. It's the longest phase. It takes time. Cells grow, prepare to divide. That's what they're doing most of the time. The rest of the cells will be dividing actively. Now we're going to talk about G1 or G0 phase. We see it on the cycle of the interphase. And characteristic for this is the G means gap. So this is they're named by the gap that follows mitosis and starts, division and beginning of the new round of DNA synthesis. So that is the gap phase, gap 1 phase. Please understand that gap 1 phase is characterized by complete duplication of organelles, complete duplication of proteins, but very importantly, there is no DNA synthesis. It will occur in S synthesis phase, next phase. Right now, cell is just making bigger, getting bigger, fatter, ready to divide. Okay, so this is G1 phase. Now, the length of G1 varies between the cells. Now, if you have rapidly dividing cells, like your red blood cells, they actually have very short G1. Boom, boom, boom. They're constantly going through the cycle. However, some cells, well, non-dividing cells are in a constant G1 phase. They're standing, they're waiting, okay? And these cells who are in prolonged G1 phase are thought to be in G0 phase, which means they're not dividing at this point. They're constantly growing or controlling their growth, but they're not going to be dividing yet. However, you can stimulate cells that are in G0 or prolonged G1 phase to start dividing if you provide them with specific signals. And these cells will now say, okay, I want to divide, and they will divide. So let's talk about the interphase or S phase. That's the next phase after G1 phase. And it's characterized by S, by synthesis of DNA, known as DNA replication. You heard about this, you talked about this, so this is just an overview. But you know that each of the 46 chromosomes in a human genome are actually copied from their sister chromatid. That's where they originate. So this is the time where DNA is being synthesized and duplicated. So how does this happen? This is again overview. You talked more in depth in this. You have ATP-dependent unwinding of the chromatin structure by the action of the enzyme of DNA helicase. Now, DNA helicase will expose binding sites for another enzyme known as DNA polymerase. You know, DNA polymerase will catalyze the synthesis of a new copy of the DNA in 5-3 direction. And importantly, you'll have multiple replication forks on different sides that will actually ensure that all genome is copied during this process. And now the newly formed chromosomes are actually packed in coiled heterochromatin inside the nucleus. A total duration of interphase or F phase is exactly 6 hours, okay? It has a constant duration. There is no variation between the cells. For every cell in the body, it requires 6 hours to do this. The next is interphase G2 phase. So it comes after the S phase, and it's a gap also phase, gap 2 phase. And it's important by this. It actually prepares now the cells for nuclear division during mitosis. So what does that mean? It's the time the cell take a breather, and then it makes sure that the DNA synthesis is complete. It says, okay, did I really divide all my DNA? Did I copy it? Am I ready to go into the separation and formation of 2 nuclei? Important for G2 phase, and we're going to talk about it in a few minutes, is that G2 phase contains a checkpoint. There is a checkpoint. There is a place where cells are checked for drunk driving. Okay, so what does it? The checkpoint is a point where actually the cells will control their DNA. They will say, am I ready to progress into mitosis? Okay, if they're not, they will be stopped. If they are, they will be allowed to progress. So total duration of interphase G2 phase is approximately 4 hours. So now it's not exactly fixed, but it's around 4 hours for every cell. Mitosis is another portion of this process, and actually it is very, very important part of the cell cycle, cell division. So mitosis is characterized by nuclear division, and it's divided into 5 phases. Now, it's rather fast. It actually takes 1 hour to do the whole mitosis process. Now, at the end of this process, what's going to happen is, and it's not part of the mitosis, is cytokinesis. Cytokinesis will be the end. Cytokinesis will be division of the cells, not nuclei, but cells now into 2 daughter cells. So we're going to go through the mitosis, and we're going to talk about different different phases of mitosis. Number 1 phase is prophase. It's the first phase of mitosis. And during this process, a nuclear envelope will still be seen. Nuclear envelope will be intact. Now, the chromatin, when these cells are in this prophase of the mitosis, is actually duplicated during S phase. But this chromatin now, we need to manage that genetic material. We can't just let it float around. So we're going to reorganize that genetic material into chromatids. So chromatids, we know, are part of the chromosome of the mitotic cells. So two chromatids will form a chromosome, and they will attach to each other at the area known as centromere, a central portion between two chromatids. Now they're going to attach on a side, if you remember cat whiskers, two specialized protein complexes, microtubules, known as kinetochores. So they're going to be attaching to the centromeres. And they will form and associate it with each chromatid. So that is actually a prophase. We see that during prophase, what's happening is we're now seeing chromosomes, as we see in this image. They're starting to form, and the cell is getting ready to divide its nucleus. So why is this significant? What's going to happen later in the cycle? You remember that later in the cycle, the mitotic spindle microtubers will attach to all of these kinetochores and will pull and rip apart chromosomes. So that is the important, that's coming next. That's not a part of prophase. But during prophase, what's going to be formed is mitotic spindle. You remember, we talked about it. Microtubules will conjoin two centrosomes and will actually form a mitotic spindle. Okay? So they will actually connect with each other. How does this happen? First, microtubules will disassemble, and they will then reassemble and reorganize to form a mitotic spindle. Now, as mitotic spindle is formed, these two centrosomes will be pulled away from each other. Why? Because these microtubules are growing. So that is the problem. That is the process. Now, during the prophase, so think about what's going to happen in prophase. You're going to have formation of chromosomes and formation of mitotic spindle. Okay? And during this process, nucleolus will disassemble during prophase. Why? Because, nucleolus is involved in the process of making ribosomes and proteins. You don't need proteins now. You need to now, all the proteins cells should have made in previous phases. It's just getting rid of nucleolus. Now the next step is prometaphase. And we see here, prometaphase is actually characterized by complete disassembly of nuclear envelope. And the next thing is that spindle microtubules will bind to kinetochores of the chromosomes. You have formed spindle, and now this spindle will actually bind to those kinetochores, to those cat whiskers on your chromosomes. And now chromosomes are now pulled by the microtubules of the mitotic spindle. Now the mitotic spindle is growing. Now think of it, chromosomes are glued to it, and they start being pulled. So this is the prometaphase. Okay? The next phase is metaphase. It's really important clinically for you. I'm going to mention to you why. But metaphase is this. As these chromosomes have been pulled, they actually will be aligned in metaphase in equator of the spindle. They will kind of form in a center. Nice line. Okay? So this is actually known as metaphasal plate. So you see here, they're all lined, nicely stacked in one line in the center. So they're aligned, and they're located halfway between two poles. Why is this process important? You remember I talked to you about the poisons like Taxol and drugs that will actually inhibit microtubules. So if you give microtubule inhibiting drugs, they will arrest the drugs in this portion of the mitosis. Okay? They will not move away from that. They will actually stay, and if you stay in the cells exposed to it, they will all have this good old metaphasal plate, but they will not go further through the cycle. The next is anaphase. Now what's happening is we're ripping apart those poor chromosomes. So we know that mitotic poles are actually pushed further and further apart. And this is actually caused by the elongation of these polar microtubules that are pushing and growing and growing. As they're growing, they will rip apart good old chromosomes. So this will result again in a separation of kinetochores that are holding the centrosomes on chromosomes. They will be pulled apart. And what's going to happen is that sister chromatids will be now moving towards the opposite side of the spindle. So you're separating them and sending them to two, like a good, car dealer in Las Vegas. It takes things and splits them in half and puts them on the side. Not that I gamble, I'm too cheap for it. But, I would say I made that money, I'm going to spend it for more stupid things than gamble. So, but it is the system. All of them are actually split into actually two sister chromatids, and they're all pulled apart and moved to the sides of the nucleus before division. Next is telophase. So telophase is this. It's the last phase of nuclear division. What happens? We are done. We rip those poor chromosomes apart. We do not need any more a mitotic spindle. So at this time, the cell will disassemble kinetochore microtubules, will disassemble mitotic spindle, and the nuclear envelopes will form around each of the two nuclei that contain those sister chromatids. They will split into two nice nuclei, okay? Now, chromatids will decondense because you don't need packaged chromosomes. You need DNA that can work. When it's packaged, it can't work. So you will now disassemble DNA, disassemble the chromosomes, and put DNA into chromatin so you can start doing normal process with the DNA, production of proteins and everything else. So chromatids will decondense into dispersed chromatin, and nucleoli will start appearing. Why? Because now new daughter cells need to start producing proteins. So they will start, nucleoli will seal, they will start producing ribosomes. Ribosomes go out of the nucleus, start producing protein. So that is the process. Now that's the last phase of mitosis. However, we're not still done yet. Yes, we have two cells that are still attached to each other, like Siamese twins. We have on the side two nuclei separate, but we need to separate the cells. So this process is known as cytokinesis. It's completion of the division of the cells. So what's going to happen? You're going to need to divide cytoplasm. Now, I hope things start coming all together from all the first lectures that we had. So what's going to happen now with this cytokinesis is this. You have two cells, okay, made in one cell, okay? And what's going to happen? You're going to develop an actin ring on a pole of the cell, like almost equator. Let's put it that way, like an equator. You're going to have a ring around actin. What can actin do? It can contract. Okay, we talked about it, cytoskeletal protein. So this actin ring is going to start contracting and contracting and contracting, which will form furrow. Furrow is cell membrane. So it will almost like to do this. It's going to contract. So this is the cell. It's contracting, contracting. Eventually, two ends meet. Boom. Cells are separate. Okay? So the contractile ring will form around the cell. It will start contracting, and two cells will separate through the process of cytokinesis. Now, interestingly, through this process, daughter cells are identical to each other, and also they're identical to the parent cell. It's really cool. It ensures that these cells are as good as their parents. Okay? Now, let's talk about Hayflick's limit. This is important. This guy actually got Nobel Prize for this. He actually looked at the cells and explored how many times normal somatic cells can divide. So Dr. Hayflick, again, got the Nobel Prize for this, said this, is the number of times the cells can divide is limited. That means that cells cannot unlimitedly divide. So depending on the type of the cell will have 15 division, 10 division, 2 division, then they will stop dividing or they will die. Okay? So that is known as Hayflick's limit. What causes the limit of this? I'm going back to Dr. Kuo's stuff, is the presence of telomeres. Do you know what telomeres are? Are those little ends at the end of the chromosomes? They're actually a hexameric DNA repeat sequence, TTAGGG. That's what they do. Telomeres are important for one reason. They ensure that chromosomes will split properly. Okay? These telomeres. Okay? So they will ensure that there is a division of chromosomes. It will occur. The chromosome will split into two chromatids. So they facilitate the movement of chromosomes to the opposite poles during telophase. But there is one problem. As we get older, our telomeres become shorter and shorter and shorter. And this will eventually result in a death of the cell, and cells will stop dividing. Okay? So that is the reason for it. However, we are super beings. We really are cool. We are, superheroes in a sense, our body. Why? Because we repair those telomeres. Okay? So telomeres we can repair by an enzyme known as telomerase. It's a complex of RNA and protein, and will help constantly add telomeres to ensure that your cells will continue dividing. But the older you get, less telomerase you're going to have, and there's going to cause a problem. So result when telomeres are gone is, or they're shortened enough, is cellular senescence or aging. That's it. That's the aging. That's the aging, actual aging process. That's why people who look for fountain of youth are trying to figure out how to invent artificial telomerases that will enable cells to divide forever. But there is a problem that can lead to cancer too. We'll show you why. So important this thing is that some tissues will require continuous cell replacements, such as skin, intestines, erythrocytes. But these cells actually originate from stem cells. Stem cells, you heard them. These are the pluripotent cells that can divide and make any other cells of the tissue. Stem cells do not care about Hayflick's limit. Neither do cancer cells. Stem cells can divide and divide and divide and divide and divide. It's very hard. They do not follow this limit. Same as cancer cells. So let's look at this. Let's talk about regulation of the cell cycle. So first, we have to understand that this cell cycle is highly regulated. And certain cell types will actually retain capability to divide throughout their lifespans, where other cells will, remember we mentioned, will permanently stay in the G0 phase. They will not be dividing. They will be very happy where they are, or G1 phase, and they will divide only when they're told to. Okay? So we know that cells that temporarily or reversibly stop dividing are viewed as in a state of quiescence called G0 phase. State of quiescence. They're just quiet, they're not doing anything, they are just surviving. While cells that permanently stop dividing, either due to age or due to accumulated DNA damage, are senescent. So the senescent cells that will not actually, they're the ones that permanently stop dividing because of their damage, they're old, they can't do this anymore. Now, very important thing is liver hepatocytes. These cells, liver hepatocytes, normally don't divide. But they have huge potential to divide when stimulated. So we can say that liver hepatocytes, cells in the liver, are actually in a state of quiescence. What does that mean is that if you stimulate them, they can actually divide. This is why this is important for you clinically. Liver transplant. Liver is the organ with the highest regenerative capability of all the organs in the body. No other organ can compare to it. So liver has a huge capability to regrow after injury or disease. I, as a kid, ended up suffering from hepatitis A from huge, epidemics that happened in a skiing center. There was a problem with water supply. Hundreds of people actually ended up having hepatitis. I'm just going on a long story. But actually it was cool because I studied that epidemics in my epidemics class in med school. So I was a part of that as a kid, going skiing, drinking water. Water was contaminated. There was some problem with water source. Cut long story short, around 1/6 of my liver remained operated. So I was in a coma for 2 months. But what happened is now if you do my liver enzymes and everything, you will not find any problem. So liver is really awesome. It regenerates, works fine. Why do you use it? Liver transplant. You literally are going to take a portion of the liver of your donor, and you're going to put that portion of the liver in the recipient. Okay? When you do this, and that was actually what I did for a living a long time ago. So when you do that, you actually remove liver and move it to another individual. So as you do this, is now what's going to happen? Your donor has less liver. Your recipient has less liver. And actually really cool, within several weeks after surgery, liver tissue will double in size on both donor and recipient. It's a really cool mechanism, so that is very, very, very important. Now, we know that most somatic cells, after a number of divisions, will enter in the state of non-division or quiescence. Why? Because, for example, if you are a cell and you divide it 30, 40 times, you're going to say, okay, now there is a better chance that I can start dividing abnormally. So these cells will go into the process of quiescence. They'll be there waiting to be stimulated to divide. So let's talk about cell cycle regulators. There are things that are constantly regulating cell cycles. So important it is, we're going to mention 3 factors that work in coordination as cell cycle regulators. Some of them are cyclins, cyclin-dependent kinases, and cyclin-dependent kinase inhibitors. So cyclins are actually cell cycle regulators that are categorized as cyclin D, E, A, and B. And different cyclins are expressed in order to regulate the specific part of the cycle. Different cyclins are expressed in phase G1, S, G2, okay? Their concentration vary during the cell cycle. And let's use a good example are D cyclins. D1, D2, D3 are actually G1 regulators. They regulate the G1 phase. So that's where they're expressed. That's where they work most. And they control the progression of the cells through a restriction point to a checkpoint in G1. Okay? They will actually make sure that if cell is okay, they will allow cells to continue. If cell is not good, they will stop the cell progressing. Okay? So there are different cyclins. There are S-phase cyclins and mitotic cyclins. There are the different cyclins that express in different portions of the cycle. What are the cyclin-dependent kinases? Cyclin-dependent kinases are a baseball bat behind cyclins. Okay? What does that mean? That means that cyclins achieve their effect through cyclin-dependent kinases, enzymes. So what do? The concentration inside the cell is constant. It doesn't change. Compared to cyclins that it changes, okay? But then what happens is that the activity of their different CDKs will vary, and the activity depends on the presence of specific activating cyclins. Cyclins need to activate to CDK for CDK kinases to work, okay? So cyclin will bind to CDK and cause their phosphorylation and activation. And now cyclins and phosphorylated CDK will phosphorylate substrates on proteins. You remember serine and threonine amino acids get phosphorylated, and that results in regulation of cell cycle. We know that CDK1 will target activated proteins that are critical for initiation of mitosis. They will regulate the entry into mitosis, while CDK2 will activate proteins that are involved in S-phase transition, that are involved during S-phase. Now let's go through checkpoints. That's my favorite part of this, because thanks to checkpoints, you know, we are healthy. So checkpoints are placed at critical points of the cell cycle, and they're actually used for cell to stop, figure out if everything is okay, and move on. It's a roadblock for cell cycle where the cells will be examined themselves whether everything is okay so they can continue. Okay? So those are the checkpoints. So they can be used to delay the cell cycle progression if necessary. If something is wrong, they can stop the cells there, give cell time to repair, or make cell kill itself. Okay? So those are the checkpoints. So cells require presence of growth factors in order to reach checkpoints. So what does that mean? You need to stimulate cells from outside to go to the checkpoint, okay? To change and grow and go to the checkpoint. After they pass the checkpoint, everything else is automatic. They do not be stimulated anymore to divide. They will just go through the cycle. So you kind of have to be supported all the way to the checkpoint. Like, so they're drunk, they're carried all the way to the checkpoints. Cops look at them and say, "You're fine." Then they drive away, no problem. Okay? So that's the idea. You know, they need to be supported by. So after passing checkpoints, the rest of the cell division always is automatic. So let's talk about tumor suppressors. Super awesome guys. Very important. So these are the cops. Okay? They're standing there on checkpoint looking for drunk drivers. So the role of these proteins is to halt the progression of cell cycle if there is a problem, such as mutation of DNA. They will say, okay, stop. Okay? And tumor suppressors proteins are also active in cells that don't want to divide. Cells that don't want to divide, they want to stay in G1 phase, they will activate the tumor suppressor, which will prevent them from moving forward, and they will stay in the G1 phase. Okay? Why is this important? What's going to happen when you have a drunken cop? Okay? What's going to happen when you have a checkpoint, then you have these tumor suppressors that are mutated and abnormal. They will let cells go. You can be drunk as hell, and they will let you go through it, okay? They'll be like, fine, go for it, you know. kind of reminds me of my own home country. So, you know, you share a drink with a policeman. Okay, it's like, are you drunk, sir? No, I'm not, but we can have some. Okay, so that's the tactics. Okay, so that's it. So like, or you ask, you know, very common thing what my friend tried. It didn't put him in jail. You know, this poor guy. He was in med school with me. He actually said to the police officer, am I adequately drunk or do I need to be drunker? And they're like, okay, now you're going to jail. So now that's the idea. He did that after passing anatomy, you know. So that was the idea, passing anatomy exam. So that was it. I don't recommend that. But there is checkpoint regulators. So let's talk about the G1 checkpoint. Okay, that's the restricting point that's found in G1. What does the G1 checkpoint checks? It checks whether cell is ready to divide. If cell is fat enough, happy enough to start going into now replication of DNA. Okay? So if the cell is ready to start going into S phase, synthesizing new DNA. So that is the characteristic of this phase. So it's going to say whether DNA is okay, is DNA ready to be replicated. Now it's going to say, is there enough resources to replicate this DNA, and is environment for division okay? In all of these things are okay, the cell will progress to checkpoint G1. So it's used by key regulating factors which make sure that this is achieved before beginning of the S phase. Now we're going to clinical stuff, kind of more interesting stuff. Let's talk about retinoblastoma. Retinoblastoma gene will produce retinoblastoma protein. And retinoblastoma protein is very important. Why? Because it regulates G1 checkpoint. It's one of those cups. Okay? It's one of the proteins, product of retinoblastoma gene, that will check whether cell is ready, does it have mutation, is ready to go towards the S phase. Now, there is a condition that they will test you extensively on board, and sadly you will see on your practice, is known as hereditary retinoblastoma. Is the condition when individuals, specifically you find it in kids, it's hereditary condition, has mutated retinoblastoma gene. So they have abnormal police officer and G1 checkpoint. Okay? Their retinoblastoma protein is abnormal. So it allows everybody to pass through the checkpoint. Now, mutated cells will go through G1 checkpoint without problem, and this will lead to increased potential for malignancies. So what happens? In resting cells, okay? In cells that are still haven't reached G1 checkpoint, okay? Retinoblastoma protein will be lightly phosphorylated. It's not going to have a lot of phosphorus on it. It's going to be lightly phosphorylated. Cells that are not yet at the checkpoint phase, okay? Now, this will prevent entry of the cells into S phase. As long as retinoblastoma protein has just few phosphorus on it, it's going to prevent entry of the S phase. How? Retinoblastoma protein will bind to transcription factor 2, also known as elongation factor, you know? So, elongation factor 2. So, it's going to bind to elongation factor when it's not phosphorylated and inhibit elongation factor. Elongation factor is necessary to induce transcription of DNA, okay? So it's going to bind, retinoblastoma will bind to transcription factor E2 or elongation factor 2, and DP1/2 factor, which will lead to inhibition of the cells. They will not move. Now, if everything is okay, retinoblastoma protein is going to get phosphorylated, okay? As the cell now reaches G1 checkpoint. Everything is fine, check phosphorylation of retinoblastoma. Everything is okay, check phosphorylation of retinoblastoma. So when a cell reaches the G1 checkpoint, retinoblastoma protein will be hyperphosphorylated by good old MAP kinase. You remember that MAP kinase that was sent? Signaling. So now when it is highly phosphorylated, it will now release the elongation 2 factor. It will just release it. Say, okay, buddy, you're ready to go. Just get out. And elongation factor 2 will now stimulate the transcription of DNA and move the cell into the S phase. Now, this whole story is important for this. So when retinoblastoma is not normal, if there is a problem, there is an abnormal function, you will have children that develop hereditary retinoblastoma. It's a very serious condition. Why? Because they will start developing tumors in their eyes. One of the things your patients are going to say, actually, I don't consider that happy occasion. I actually helped one of my friends diagnose their kid with retinoblastoma. So what they do is they show the kid's pictures. They're going to say, "Doc, this is cool, but my kid just has one red eye. The other one is white on all the pictures." That actually tells you that this kid has retinoblastoma tumor, okay, in their eye. So it's actually caused by mutation of retinoblastoma, and symptoms with white color in the center of the pupil when light is shown in the eye, such as taking flash photography. And eyes will also appear to be looking in different direction. They will be swollen. But usually you don't see much. You just see that white spot. This is the problem. So some of the children, especially those in which the tumor involves both eyes, so it can actually involve both eyes, are in the risk for developing other tumors, such as osteogenic sarcoma, very bad stuff all over their bones. So if they have retinoblastoma problem, they will have problems with their eyes, but they will also develop tumors of their bones. So interesting for this. It's kind of interesting for you to know, okay? Retinoblastoma you will never expose to radiotherapy, okay? It's interesting. This is one of the rare tumors. When you expose them to radiotherapy, they break apart and disseminate, okay? So you don't want to disseminate this tumor. So most of the time, whenever there is a tumor in place, you do not subject these kids to radiotherapy. So what's the therapy? Therapy is removal of the eye. So you want to stop that cancer progressing. Now you can give them radiation therapy because tumor is gone, and chemotherapy. And if it is not discovered early, can definitely significantly decrease the lifespan of the child. So let's talk about my hero. You know, if somebody likes Superman, Spider-Man, or everything else, this is my hero, okay? This is a protein, tumor suppressor protein P53, kind of awesome. This is the tumor suppressor protein that plays a role in regulation of G1. As retinoblastoma plays a regulation in G1, he also plays a regulation in G1, but he has superpowers, okay? He's really cool. Why? Whenever is a nuclear damage, whenever is nuclear damage in the cell, there's a problem in the cell, this will result in phosphorylation of P53. Now, P53 will now activate P21, another protein, and will stop the cell. So if there is a mutation of something else, P53 is going to get phosphorylated, activated. P53 will tell P51, stop everything, okay? And let's repair the damage. Let's repair that mutation. Cells will work to repair that, damage, but if the repair is inadequate, there's really serious mutation, then it's going to tell cells to kill itself. So it's going to progress to apoptosis, it's going to have stop the party, pull a gun in your head, goodbye, okay? So that is what it does. It stops cancerous cell. It's really cool. So if damage is irreparable, P53 will trigger apoptosis. And there's a clinical significance, like mutation of P53 will lead to uncontrolled division. What's going to happen when P53 doesn't work? Boom, you're getting a lot of junk going down the cycle, okay? And the problem is that P53 mutation is found in half of all cancers, human cancers. So it's really, really important. Why is P53 important? There's a lot of discussion about vaccination for cervical cancer, okay? Cervical cancer, some people say, people get very affected by the fact that we are vaccinating young girls in their, 10, 12 years old for a sexual transmitted disease, okay? And parents get really upset about it, obviously. They're like, oh my God, it's 10-year-old. You're giving them, papilloma virus is transmitted through sexual contact, okay? But sadly, more than half of US adult Americans have they're papilloma virus positive, okay? So that's a problem. So vaccination, so men can be okay with papilloma. They might develop weird warts that don't look very pleasant, but women will end up with cervical cancer, okay? So that's why it is. And what papilloma virus strains 16 and 18 are actually associated with cervical cancer. So we can give vaccines against this. So what is it? This viral protein that's produced by this papilloma virus, E6 will bind to P53. So to produce a protein that will inhibit P53. This virus will also produce protein, double whammy, that will inhibit E7, that will inhibit retinoblastoma protein. So now G1 checkpoint, no more, okay? everything goes through G1 checkpoint, okay? And this will result in development of cervical cancer. This is how papilloma virus works. It's extremely, extremely contagious. So it's not something that you would say, oh, you know, it's an STD. Yes, it is, but it's actually extremely contagious. It can be carried, compared to a lot of stuff, through even objects. As every virus can remain for a longer time. Let's talk about G2 checkpoint. It's an important checkpoint that occurs after the S phase and before the M phase. So G2, remember, S phase done, M phase, G2. We mentioned this already. And it is used to confirm that DNA is completely duplicated before the beginning of mitosis. You know, DNA is duplicated, and G2 checkpoint, they check, go with a flashlight, say, okay, yeah, you know, all the DNA is duplicated. Move on, okay? It all looks fine. So that's a characteristic, and that's the period where CDK1, cyclin-dependent kinase 1, plays an important role. It controls the entry into the mitosis. So how does it work? During G1, S, and G2, CDK1 is phosphorylated on tyrosine residues, and it is inactive. So what does that mean? Is that guy that controls, it's inactive. If it remains phosphorylized, cell will not go through the G2 checkpoint. So what's going to happen? So if everything is okay, this is what's going to happen. An enzyme known as CDC25C, if everything is okay in the G2 point, will dephosphorylize CDK. It's going to say, okay, guys, everything is fine. Let's dephosphorylize CDK1. CDK1 will now bind to cyclin B, which will now enable cells to go through the phase 2. Okay? So during the process, it's constantly phosphorylized. If everything is okay, the CDC25C phosphatase will dephosphorylize CDK1, and CDK1 will now bind to cyclin B and move the cells further. Now, this is also important. Let's talk about what's happening in different situations. So you have DNA mutation. This will cause the arrest of cell cycle in G1 phase. And arrest will last until DNA is repaired. We know this. If there is a problem, cells will stop in the G1 phase, and DNA will be repaired. And when everything is fine, P53 will say go. Okay, you're fine, you're ready. But there are different responses in the cells depending on what type of injury is causing it. Okay, there are different responses. So, for example, ATM, ataxia-telangiectasia mutated protein, will respond to double-strand DNA breaks and ionizing radiation damage. They will try to actually respond to that. That's how they're going to detect the damage. How do you know something is wrong? The ATM will detect it. You know, the place where you get your money from. So, it responds to double-strand DNA breaks and ionizing radiation damage, and it will be activated. ATR will respond to UV-induced DNA damage that we already mentioned. Now let's talk about response of S-phase cells to DNA damage. There is also an S-phase checkpoint. Why am I not talking about S-phase checkpoint? Because it's rather complicated. Now the story goes even more complex. Why? You remember what's happening through the S-phase? DNA is being constantly doubled. Okay? And during this process, we have to ensure that there is no DNA damage during this process. Okay? If there is a DNA damage, the whole S-phase will slow down and give time for cell to repair the damage. Okay? So that is the S-phase. Important player here is BRSA1. I don't know if you heard about BRSA1, but BRSA1 is actually protein product of breast cell susceptibility gene 1. It is a very important normal protein that actually regulates the progression through S-phase, through S checkpoint. However, if it is mutated, such as individuals who have higher potential to develop breast cancer, this S-phase checkpoint will not work. So the individuals who have BRSA1 mutation, you can detect in them, have higher risk of cancer. Guys, for your next lecture, you're going to realize that. Don't be Angelina Jolie, okay? Why? If you have mutation of BRSA gene, that doesn't mean you're going to get breast cancer. It means that you have higher chance of developing breast cancer. So don't go off hacking parts of your body, okay? Because it's going to look good on TV, okay? So what's important is if there is an individual who has family history, who has BRSA1 positive, these individuals have to be followed, Okay? It doesn't mean they need to do anything unless they start getting positive tumors. Now, thank God, due to the advance of the surgery, another thing that I really was doing long time ago is you will remove that lump. And if the lumps become more concerning, then you're going to remove the breast. But, you know, if you just want to go Angelina Jolie and say, I have Bursa 1, go for it, buddy. Take everything out, okay? So what it is that is not, and I'm going to explain to you in the next lecture why. You need to develop and accumulate several mutations in order to get cancer. It's not just one. So let's talk about anti-cancer drugs. So both tumor and normal cells will undergo the same cycle. We know this. So are anti-cancer drugs good? They attack your cell cycle? Of course not. You know, they will affect your cells too. So why do you give your patients anti-cancer drugs that will block the division and stop the cells from dividing and actually cause them to die? One catch. Why does anti-cancer medication work? Because percentage-wise, more of the cancer cells are in the active phase of division than your normal cells. Does that make sense? Because cancer cells are going like crazy constantly through this cycle, while your cells are taking their sweet time. Okay, so they're checking everything. They're checking G1. Okay, let's see G2. So if you go with the cell-specific, for example, anti-cancer drug that affects cells in a phase S, okay, it's going to have more chance of affecting cancer cells. There's going to more chance to affect yours. Your cells will be collateral damage, but damage will be less. Okay, so that is the idea. That's why. So anti-cancer therapies will utilize that. There are cell-specific anti-cancer drugs. So there are anti-cancer drugs that actually target specific cell portion of cell cycle. And you're going to give these drugs to whom? You're going to give these drugs with to patients who have cancers with high rate. You know why? Because there is better chance that cancer cells are in S phase than your normal cells. So you're going to give a drug that affects cancer cells and stops everything in S phase. Okay? Because now these cells are much highly rapidly dividing, so it's a better chance to catch more cancer cells in S phase. It's like fishing. It's like fishing, okay? You destroy them. However, if you have tumors with low division, they're the problem. Why? Because they might be dividing at the same rate as your normal cells. Now, these are very potent cancer cells. They will stop all the cells from dividing, you know, specific groups of cells. But you have to use those. They are non-cycle specific. They do not work in specific portion of the cycle. They will work throughout the entire thing. It will stop the whole thing. So the example is anti-metabolites, which are the low division rate. What are the anti-metabolites? Anti-metabolites are cancer drugs that look like normal stuff. They actually look like your purine or pyrimidine, okay? And they're standing and waiting to be picked up, you know, during transcription. So when they're picked up, they go into the DNA and block everything. Kaboom. And cause a fraction of the DNA. And that causes cell. P53 goes nuts, say, oh my God, the cell is not normal. Stop everything, kill it. Okay? So that actually helps you. So that's the idea. These are antimetabolites. Good example is 5-fluorouracil. It's actually methotrexate, which is a drug that looks like one of the bases in your DNA. And it's going to be put into the DNA and will cause problems. It's also used as a morning after pill because of that effect. It will stop a division of the cell. Now, anti-cancer antibiotics, I love them because that was my area of research with apoptosis too, these are actually drugs that will accumulate in a G2 phase of cycle. So, like a bleomycin. However, most of the anti-cancer antibiotics are not cell cycle specific. So where are you going to give the anti-cancer antibiotics to? They're not cell cycle specific. Slow-growing tumors. These are usually pediatric tumors. Okay? It takes for them time. They're very toxic. Okay? But it's a kid. Okay? They have much better chance of regeneration. And they will actually affect all of the cells. Because the pediatric cancer cells are going through cycle rather slowly. Okay? So they actually will directly or indirectly interact with DNA and disrupt its function. So they're used to treat solid tumors with low growth fractions such as pediatric tumors. I actually work on a drug known as daunomycin or adriamycin, which is a fantastic drug. And it's awesome drug. It solves all the problems. However, it kills certain population of patients from heart failure in 10 years. So, you know, you give it to patients as kids, and, you know, they're fine. They actually get rid of their cancer, and then 15 years later, they come for heart transplant. So now this fantastic drug is actually being underutilized because you have to give lower doses for them not to develop a heart problem. So the idea on my lab and my research was how to stop that. So how to be able to stop the bad effect of daunorubicin on heart and at the same time enable it to kill a cancer. And actually we were successful, but there was no economic interest to continue that from a side of pharmaceuticals. But you're going to hear that more. Now, next thing is mitotic spindle drugs. You know this. These will inhibit specific cell phase. They inhibit M phase. You remember mitosis, where when the mitotic spindle is formed, when they form the plate? Okay? So these are cell cycle specific. So where are you going to use them? You're going to use them with rapidly dividing cells because they're going through a cell cycle. You can stop a specific phase. Okay? So this we mentioned already. These are microtubules or mitotic spindle poisons. Okay? Okay, guys, I'll give you a little break, and then we'll be back for our next lecture. Thank you.